and say welcome Bingyu Yan from Majid Kazemian's lab in the Department of Biochemistry here at Purdue University. Bingyu, I've been wanting to get you to tell us your journey because you have a similar one to me, where you have a plant biology background and now you've been doing this great work in looking at immunology and SARS-CoV-2 and all of this wonderful biomedical work that you're getting into. So it's a pleasure having you on, and thank you so much for coming to our Purdue Lecture Hall series. And again, it's awesome having you on the program.
Thank you so much, Tommy, for the introduction.
Awesome. Well, so I'll tell you what, if you don't mind, you can share your screen, give us a little bit of your background, what research you're doing and all that good stuff. And I'll turn myself off. And then at the end, if you don't mind, I'll come and we'll talk a little bit more. How about that?
Sure, that sounds good. Let me
Perfect.
share my screen. So can you see my screen?
I can. Perfect. Thank you so much.
Okay, thanks. So now let's get started. Thanks for having me here. I'm very glad to introduce one of my projects I've done during my PhD. So the topic for today is we found SARS coronavirus 2 could drive JAK-dependent local complement hyperactivation in the COVID-19 patient lung. So we know the SARS-CoV-2 infection causes a pneumonia named COVID-19, and the patient would got hyperinflammation and tissue damage in their lung. And the severe cases are resulting in death. So here I'm showing you the current situation about this pandemic. So until now, the global situation is there are more than 700 million confirmed cases. So here I'm showing the bar plot, recorded the weekly confirmed cases over the 3 years. And also there are about 7 million deaths until now. So in March 2020, at that time, there were more than 100,000 cases over more than 100 countries, and there were more than 4,000 deaths at that time. Then the World Health Organization declared COVID-19 as a pandemic. So we start our project about the same time in March 2020, and we were curious about what is happening when the patient get infected with SARS-CoV-2. So basically, we want to investigate the cellular response to this virus infection. So there are 3 questions we want to ask. The first one is, what are the biological pathways that are induced by SARS coronavirus 2? The second question is, what are the regulators and what is the mechanism that driving this change? And the third point we want to seek is whether we can find some existing drugs that can be used as a treatment. So to answer the first question, I sourced all the publicly available gene expression data at that time. So remember, at the beginning of the pandemic, the SARS-CoV-2 infected samples and data are very limited. So I found a data set which quantify the gene expression in SARS-CoV-2 infected cells. They are the A549 cell, which is a long epithelial cell, and A549 transduced with ACE2, which is an entry receptor for the virus. Also, there is a primary human epithelial cells that are got infected with SARS-CoV-2, and they measure the gene expression. More importantly, there are 2 COVID-19 patient lung biopsies got quantified with their gene expression data. And with all this publicly available data, I'm able to compare SARS-CoV-2 infected samples versus non-infected or mock infected samples. then find the differential expressed genes and pathways. So let's take a look at the results we got. Here I'm showing you all the SARS-CoV-2 induced pathways. And among all the 4 data sets I mentioned, there are 14 pathways that are consistently present and shared by them. So by carefully checking all these pathways, we found some of them are typical virus infection response pathways like interferon producing and some of the immune activation. However, 4 out of these pathways are complement associated. And what is complement pathway and why it is important here? So let me provide a brief background here. So the complement pathway is a evolutionary conserved pathway that responding to infection and then activate the inflammatory response and clean pathogens. So there are 3 different routes that can activate this pathway. So all these 3 routes ended up with activating of the C3 convertase, and the C3 will be cleaved into 2 active forms, one called C3A and another called C3B. The C3A is a C3AR ligand which can activate myeloid cells and promote inflammation. The C3B is a CD46 ligand which can activate downstream lymphoid signaling to promote the immune activation. So here we can see the C3 is at the hub of the pathway, and which means it is a key component here. So in general, this pathway is protective, but when it got hyperactivated, it can be pathogenic. So this pathway would drive pro-inflammatory signature in the lung and the tissue, or cause tissue damage. So initially, people believe this complement system is basically a serum-based system, which means the complement factors are generated by the liver and distributed through the bloodstream to work on other tissues and cells. But remember here, we found when the lung tissue are infected by the SARS coronavirus 2, it starts to produce or activate the complement pathway. So it indicates the complement could be present and active intracellularly in lung. To test this hypothesis, I checked the SARS coronavirus 2 infected cells by comparing with uninfected control. We can see all the highly induction of complement genes. Here, the complement factor B, C3, which is a key component for this pathway. And by checking the transcription of this C3 gene, we can see the SARS coronavirus 2 induces this C3 transcription in different lung cell lines. So with this data, we want to know whether the complement is at the protein level is activated in the lung cells intracellularly. To test this hypothesis, we set up a model. We infected the cells with SARS coronavirus 2. On the top panel, I'm showing you the mock infected cells, which we stained the nucleocapsid protein of the SARS coronavirus 2. And then here in the mock, we don't see any infection of the virus, and there is no signal of the C3A. However, if we infected the cells with this virus, we can see clearly these cells are present some nucleocapsid protein staining, and we can clearly see the induction of the C3, which means the complement is activated intracellularly in these infected cells. By summarizing all the data here, we have a quantitative results. We can see the infected cells have a higher induction of the C3A by comparing with uninfected cells and mock infected cells. Additionally, we also found the coronavirus viral load has a very nice positive correlation with the C3A intensity in each cell, which means if you have more virus in the cell, the complement activation would be higher. So here, from this data, we could conclude the C3 now is intracellularly induced and activated in lung epithelial cells. And our next question is, we want to know whether in the lung tissue microenvironment from the COVID-19 patients, the complement is get activated in other cell types, for example, the myeloid cells and lymphoid cells. To investigate this, I sourced a publicly available single-cell RNA sequencing data from the COVID-19 patient lung sample. By processing the data and got the gene expression, here I'm showing you all the cell types I got from the COVID-19 patient. So in this UMAP, each dot is a single cell, and the cells are clustered together based on their gene expression similarity. And by annotating the cell types with canonical cell markers, we identified 3 major cell types from these patients. So there are lymphoid cells, myeloid cells, and epithelial cells. And here are the detailed cell types annotated. So with all this single cell gene expression data, I could identify cell types and then calculate the complement pathway activity in different cells. So by doing so here, I'm showing you the pathway activity score I got. So on the top panel here, I projected the score on the same UMAP. And if you see a cell with a darker color, it means the pathway activity is higher. The first pathway I showed here is CD46 regulated genes, which belongs to the complement-induced pathway in lymphoid cells. By comparing with the uninfected lung, we can see a strong induction of this pathway in COVID-19 patients, both in mild and severe cases. I also checked another pathway called C3AR1 regulated genes, which are belonging to the complement-induced pathway in myeloid cells. We observed a similar trend, the uninfected lung and the induction of this pathway is higher in the mild and severe COVID-19 patients. So with this, one more point I want to add here is we found the complement pathway activity could track the disease severity very well. Here, as this violin plot shows, in the lymphoid cells, we see the CD46 regulated genes, their activity is going higher when the disease are getting severe. Same observations is true in the myeloid cells for the C3AR1 regulated genes as well. So for now, let me summarize the first part is we found upon SARS coronavirus 2 infection, the complement pathways are activated intracellularly in lung. And we found the C3A is positively correlated with the viral load in the cells. And we found the complement signature in the COVID-19 patients track the disease severity very well. And next, we move to our second question is, what are the regulators that driving this complement activation? So to answer this question, we predicted the upstream regulators of the SARS-CoV-2 induced differential express genes in a gene regulatory network. So here I'm showing you the results. So the top regulators are here the JAK-STAT factors, which are highlighted in red, and also the NF-kappa B associated factors, which are highlighted in blue. So our next question is whether this factor do directly regulate the complement genes. So we take a look at the binding of these transcription factors to the promoter region of 2 complement genes. As an example, here the complement factor B and the C3 genes. We clearly see these genes are activated in lung by the A3K27 acylation mark. And for sure, the STAT1 and REL-A is bind to the promoter of these genes, which means the complement genes shown here are direct targets of STAT1 and REL-A. So after solving the mechanism, our third task is we want to find a drug that can be used as a treatment to normalize the SARS coronavirus to induce DGs in the lung cells. So we built a model to predict all the FDA-approved drugs, which of them can normalize the DGs induced by SARS coronavirus 2. So our top hit here is called ruxolitinib. After reading literature and check the information about this drug, we found this is a JAK-STAT inhibitor, which is matching our previous regulator prediction. So I checked another data set, which infected the A549 cells with the SARS coronavirus 2. You can see clearly at the transcription level, these complement genes are induced. And if the ruxolitinib is treating this SARS coronavirus infected cells, we see all these complement associated genes are normalized by this drug, which means the ruxolitinib is able to normalize the transcription of the complement associated genes in lung cells. So with this data, a hypothesis, the ruxolitinib is able to normalize the complement hyperactivation at protein level in lung cells that are infected by SARS coronavirus 2. To test this hypothesis, we set up an infection model and treated the cells with either complement inhibitor ruxolitinib, which is our predicted top hit, and we have a third testing for ruxolitinib plus remdesivir, which is an antiviral drug. Remember, we found the viral load is correlated with the active protein C3a in the lung cells. So after the treatment, what we found is the complement factor B inhibitor is efficiently suppress the complement activation in the lung cells. Same observation for the ruxolitinib, we see a effectively downregulation of the complement activity in the cells. And if we combine the ruxolitinib with remdesivir, we can see the complement activity is bring back to uninfected cell level, which is very promising. So now let's summary this story. We found in this project, we found upon SARS coronavirus 2 infection, the lung cells are activate the complement genes through the interferon JAK-STAT pathway by cooperating with NF-kappa B factors. So the key component C3 is cleaved to two active forms, C3a and C3b, to activate downstream immune cells, then cause tissue damage and inflammation. We predicted a treatment strategy for the COVID-19 patients is to use complement inhibitor, antiviral drugs, and JAK-STAT inhibitor in combination with proper dosage. And we want this would be helpful to relieve the tissue damage and inflammation in the severe COVID-19 patients. So if you want to know more about this story, you can scan this QR code to read our paper published on Science Immunology in early 2021. So that's the what I want to tell you about my research for today, and I'm happy to take any question. And with this, I want to thank my advisor, Dr. Majid Kazmi, who guided this project, and also our collaborators, Dr. Claudia Kemper, Dr. Ben Afzali from the NIH, and also Dr. Christian Wobas from the University of Michigan. So this work is really a team effort, and I want to acknowledge all the co-authors who contributed to this paper. And after this, I just want to give a brief introduction about myself. So I'm from China, and I'm a PhD student in the Department of Biochemistry from the Interdisciplinary Life Science program. And before I became a PhD student, I did a master in China, which I investigate cancer evolution and also some evolutionary biology. So during the past few years in Purdue, I got involved in different projects which are focusing on investigating host pathogen interactions, as I shown today here, how cells are responded to SARS coronavirus infection. And also I have projects on virus-associated cancer. And my PhD thesis project is try to investigate the enhancer RNA's function in immune cell differentiation. So that's briefly my background, and I'm happy to take any questions now.
Awesome. Thank you so much. That's really good. And I wonder if you can stop sharing your screen, and like that we can be bigger on the presentation. There we go.
Sure. Thank you.
Thank you. Thank you so much for that fascinating talk. You know what it is so amazing is how simply you just you were dealt into a lot of complex information, and it's a lot of data driven type of information. Maybe I would ask if you were to explain it to, a high school student, what is the main gist? What should we know about the results of your latest paper?
Yeah. Let me do it this way. So in this work, because we started early in the pandemic time, so we want to find, okay, what is the virus change the cells and then lead to a severe symptom in the COVID-19 patients. So, and all this could be quantified from the gene expression of the lung cells that are infected with this virus. When you compare the infected cells versus non-infected cells, you do see some of the genes, they are bring up by the virus, and that might be associated with the disease severity. So we did in this way, and we found the complement system is got hyperactivated in the severe patients. And when we know this, we want to briefly knock down this hyperactivation. And how we can do this is we found some drug which can repress the genes that are induced by the virus. And then we are trying to bring back the cells to a normal status and relieve this inflammatory signature.
That's awesome. That's perfect. And what's incredible is, again, this data-driven approach. Like, where do we find the data? A lot of the data is already out there. Can you explain a little bit about, what are some of the tools that you use to do the mining of the data, to do the statistical analysis and, pattern finding and significance, all of that good stuff?
Okay, yeah, that's a great question. So basically, all the data is available on the NCBI website. So different groups of scientists, they measure the gene expression from samples, and then they release their data on the website. And then you can just basically go there and find what you want by searching it. And then you download the data. But when we got to the analyzing part, we do need some coding and programming, which is to quantify gene expression data from high throughput sequencing. And then we build a table for the expression value of each gene, and then we can compare. And this includes some statistical analysis to find which are really different between uninfected samples and infected samples.
That is awesome. But so then the data is publicly available? Anybody can get to this data?
Yeah, it is publicly available. Yeah.
That's awesome, because I don't think people, like, maybe I don't know if high school students know that this work that we're doing in science, we're giving back to the public as publicly funded per se, government and so on, companies too, but that, we're giving back this information so that it is publicly accessible, so that, we can work on it, and we can look at the different patterns, as I said before, and so on. But that's so fascinating. So, Bingyu, tell us a little bit, your story in terms of like when you came from China. What were you expecting? What were you anticipating how it would be? And maybe what were some of your surprises?
Um, Actually, I don't have much ideas. When I first came here, I just want to polish my self on research area and find the things I'm interested. So luckily, I found Dr. Kazmian's lab, and he provided me the environment to learn different skills and discuss ideas with him. I think initially, I think I'm getting my PhD and get trained on how to be an independent scientist. And I think now I'm approaching there.
Awesome. Awesome. And what does it mean to be an independent scientist?
Um, I think that's really a big question. So for me, is I want to investigate the questions, to ask different questions in the area of immunology. So for example, how immune cells kill cancer cells, and how the pathogen infection changed the microenvironment for the immune cells, and whether we could provided some drug prediction to promote immune function. So being an independent researcher provided you the environment to do this and solve the problem you are interested. So that's what I'm thinking about this
Fascinating.
question.
Fascinating. I love it. And so, can you recall like what inspired you what inspired your interest in immunology? Is there any particular thought or event in your life that you said, "Oh, I really would love to study immunology"?
Um, so the start point of this is I want to learn cancer biology and try to find a way to help the developing the treatment for cancer.
Yeah.
And during my career, I found, okay, the immune therapy could be a promising direction to go. So because we could boost our immune system to combat with the malignant cells in the body. So then I focus a little bit more on the immunology part, and I found it's a fascinating area, and it is complex, and we could identify different factors that can promote this process and then treat cancer.
Yeah. Awesome. Awesome. And what would you say would be good courses for people to take if they were interested in doing some of the work that you've done? And I love that you put the QR code for the publication. That's such a great idea. I love it. I'm going to ask people to do that from now on if they can. But yeah, so what type of courses, what preparation is good to have for doing this type of work?
So there are two parts for driving this work. The first one is the analyzing of data. So for this, you need to know how to handle a big amount of data on clusters like supercomputers. You need to learn some programming, statistics, and data structure courses. And another part for this is the biological part, which you need to know some knowledge in genetics, molecular biology, and also you may want to choose a specific direction you want to go, like immunology, cancer biology, or plant science.
Yeah, that's wonderful. And you're in the Department of Biochemistry,
Yes.
which brings together all of these fields into one department. So you have plant biologists and human and animal biologists and biochemists doing all kinds of interesting work. What was it? Do you remember, if you recall, what was it that got you interested in science? Do you remember what it was, the curiosity of science?
Oh, yeah, that's a good question. So when I was young as a child, in primary school, I was fascinated about plant. As I mentioned, like I was very interested in plant biology because the flowers and the different plants, the uh, have different morphology, like color of the flowers. So at that time, I was super excited about that, and I want to know, like, okay, what drives this difference? Because for doing science, important point is to ask questions. So then I think this curiosity to the difference in plants, and then this drives my very early primary ideas in doing this.
Yeah, no, for sure, for sure. And I think I recognize that in looking at your background, that you started with this plant biology track. And tell us a little bit what it was, how was it to change from plant biology to now more human biology, or biomedical type of questions?
Oh, okay. So the story is when I start my undergraduate training, I was in a lab which doing cancer evolution and evolutionary biology. And at that time, I got trained in biochemistry, and we are doing some drug development. But in the meantime, I got involved in a plant science lab, which which they are investigate how to use a new technique called CRISPR to regulate genes in plants to fulfill some pathogen-resistant phenotypes in the plants.
Awesome. Awesome. So you've been able to use some of that knowledge in your current project too?
Yes, in my thesis project, we applied some sophisticated technology like CRISPR to try to regulate the genes in the immune cells.
Yeah.
Yeah, but it is the like basic knowledge from the molecular biology and genetics, and then drives me switch my uh, research direction to the human related stuff after I got trained in both area.
So tell us a little bit about this CRISPR technology, and maybe if you can explain a little bit, what is it and how is it different than what we were able to do before with just genetic engineering?
Okay, yeah, that's a great question. So the CRISPR system is a immune system found in bacteria, which they can combat virus infection. So the specificity of this system is very fascinating. So basically, you can get the Cas9 protein to anywhere in the genome which has a PAM sequence, and then the Cas9 nucleus will cut there to generate a double strand break on the DNA. So previously, when people doing this, I think before this CRISPR and TALEN techniques, so we cannot do a specific targeting to a genomic area. But this technique do enable us to like knock out a pieces of DNA specifically from the genome in different species.
And we can do it just one nucleotide, right? We can just mutate one nucleotide or knock it out or remove it or edit very, very discreetly, can't we?
Yeah, for now, the technique is developing very fast, and we can do single nucleotide modification by using the CRISPR related techniques.
Yeah, it's so neat. It's so neat and it's so awesome. So tell us something. You talk about this data piece, in
Mhm.
your life, in your lab. And there's also this biology piece. Are you in the lab doing biological assessments and doing, PCR analysis and so on? Are you actually culturing cells and looking at their biology as well as doing data sciences on the side?
Yes. So I can work on both sides, like the data science part and also the biology part. But it depends on the progress of the project.
Yeah.
So basically, we start from mining big data, and then we have our hypothesis, and then we test it if we want to use some experiments. So I can do PCR and cell culture in the lab, yeah, which I feel is very exciting because you can probe into different area and try to answer the question from different angle.
And it's so amazing to have somebody like you that has both talent in the data side and management, manipulation, analysis, but then the biological side too, and being able to bring these two forces together is awesome, because it's, as you know, it's very rare that a computational scientist can talk clearly with a biologist, and you bring both of these worlds together.
Thank you.
And it's great. Bingyu,
Thank you so much.
I have a question that
Mhm.
I want to ask. If you were to give recommendations for students from China that want to come and do research here in the United States, what are some of the things that they should do to prepare? What are some of the things that you have found that were helpful in your preparation to come?
Um, yeah, from my perspective, I think many of Chinese students, they are very well qualified in doing research all over the world. But to my cases, when I first came here, I found uh, my English, the language, is kind of a point that I want to improve a lot, because it's not only for the daily communication with people, but also, the science is a team effort. You need to communicate your data and your results and ask questions with different people. So I think, from my perspective, I think the language and communication is a very important point for Chinese students or all the other international students.
And so what, did you take a course in English, or did you take technical writing? What how have you prepared, or what have the techniques that you've used?
Okay, so here I think at Purdue we have great resources. In the first year, I took a class they teach English communication. Basically, it want to train us to qualify the teaching as assistant position. But from that class, I learned a lot to for how to communicate in English. We form small groups to communicate in English and prepare presentations. I think that's a very nice class. And also, I went to the Purdue Writing Lab several times, and they provide very nice suggestions on how to modify your uh, like proposals and how to efficiently communicate your idea with people. Yeah, I think we have great resources here. I think people should like take use of it.
Yeah, yeah. And I also think it wouldn't be a bad idea to recommend that people start, even before they come, to start taking some classes and getting more comfortable with it. The other thing is, and I'm also international, meaning English
Mhm.
wasn't my first language. I also think that it's good to mix in with, as you say, it's a team effort. So mix in with with the lab and mix in with the other people in the lab that have no choice but to only be able to communicate in English, right? It's
Yeah,
and force yourself to get out of your comfort zone culturally and in the language part too, so that you're forcing yourself to speak more eloquently or speak more frequently with other people, and so you'll have to communicate more. Um, there's this aspect too of, you know, the other people in the lab teaching you techniques, teaching you ways. Is it always up to you, Bingyu, or did you like rely on other people? And you did such a nice job thanking everybody
thanks.
in your publications and also thanking your collaborators and the people in your lab. Tell us a little bit about the dynamic there.
Yeah, I think the research and science really, as I mentioned before, is a team effort. So different people, they have different area of expertise. So I think for scientists, you need to understand what is going on in this project, but I don't think everybody like can sophisticated in every single area. So with that, we need to collaborate, which means we integrate the expertise from different people and push the project forward. So in the lab, I think our lab and our collaborators, we are highly collaborative. So we provided hands to different people, and during this process, you could learn from people with different background, and then promote your own like build up of your like expertise.
Yeah, absolutely. And you go together, right? You
Exactly.
help each other out. I love that. I think that that's really great. So what's next for you? What's coming up next in your journey, in your adventure?
Um, my next position is I will be a postdoc fellow at Stanford. So to investigate more on the gene expression regulation from the non-coding RNAs, and also like I want to use immunology as a system to study these questions, and also later on I would be very interested in to develop some RNA therapy in cancer treatment or autoimmune disease treatment.
Awesome. Awesome. So I should really be congratulating you on passing your PhD defense and really calling you Dr. Yan.
Thank you
So
so much.
thank you, Dr. Yan, so much for giving this wonderful lecture and letting us peer a little bit closer into this point of the complement system and its hyperactivity during the SARS-CoV-2 infection. What a wonderful, wonderful way to lead us into this journey and giving us your guidance as you've come all the way from plants to do this wonderful work. So best wishes to you in your postdoc, and thank you again, Dr. Bingyu Yan from Biochemistry Department here at Purdue University, working with Dr. Majid Kazemian. Thank you so much.
Thank you, Tommy, for having me here. I really appreciate this opportunity, and it's my great pleasure.
Well, it was our great pleasure, and hope to see you around again. Take care and
Sure.
have a great afternoon.
Thank you. You too.
Thanks. Bye-bye.
Bye bye.